it's great to be with you here this morning. As David mentioned, my name is Megan McCaghey, and I am a relatively new professor in the Department of Plant Pathology in the Twin Cities. I started just over a year ago, so I'm going to start off just by giving a very brief introduction as to who I am and the type of work that I do. I'm then going to talk about my soybean projects that I've had over the past year in my first field season, and I'm also going to discuss work conducted by Ashish Ranjan, who is another faculty member in the department who was not able to join us today. All right. So at Minnesota, my research focus is on soil-borne fungi that cause crop diseases. So there are a variety of fungi and oomycetes that I'm currently working with. And overall, my program is focused on improving management to diseases caused by soil-borne fungi by considering the ecology and epidemiology of pathogens. We're also working to enhance host disease resistance, which is going to be the focus of today's presentation. So my position is research and teaching. I don't have an extension appointment, but my position is funded by AGREET, which is a consortium of growers and industry representatives. So there is an expectation that I do work that will benefit growers. And ultimately, that's what really interests me in plant pathology research. I want to take questions that are developed in the field and use a variety of approaches, both basic and in the lab, and applied and in the field, to answer those questions. So I am interested in learning more about challenges that you experience as growers in ways that we can work to address those challenges. So I would encourage you, if you have ideas or thoughts, please feel free to reach out to me. My email address is here on the projector, and my telephone number is listed there as well. And I'll be around the conference today and would enjoy the opportunity to talk with you if you have ideas. All right, so Angie already introduced sclerotinia, which causes the disease of white mold in soybean, and I'm sure many of you are too familiar with this disease. But the important aspect of scouting for this disease is looking for these small mushrooms called apothecia. So part of the reason sclerotinia is so difficult to manage is that it forms these hardened resting structures called sclerotia. And we like to refer to sclerotia in terms of looking for them in the field or on your plants as structures that look kind of like mouse droppings. So they're hardened, small, black structures. And when conditions are cool and moist for extended periods of time, this small mushroom called an apothecia will form. And there are some lookalikes of this mushroom in the field to be aware of. So there's another fungus, another mushroom called bird's nest, which also has a cup shape, but it has kind of darkened round structures inside of it that almost look like bird's eggs. There are also mushrooms that have, instead of a cup shape, an umbrella shape. So this is distinct in that it has this cup shape, and it's quite small. It's about the size of an eraser head, but it can vary a bit. So the reason that it's important to look for this mushroom is that they are what release the ascospores. And the important period of infection for sclerotinia is at early flowering stages. So in terms of fungicide applications, getting those preventative fungicide applications on your plants between R1 and R3 is the very important window. So these ascospores are released onto flowers, and this fungus is really good at breaking apart the flowers and getting at those nutrients within the flowers. It is then able to progress down the flower to the main stem, where it causes the lesions that are going to result in yield loss. So this is a very destructive disease, as you may be aware. So in this field, you can notice some brown patches throughout. And when you walk into the field, we're able to notice classic severe symptoms and signs of white mold. So these include bleached lesions on the main stem and poor pod fill. One nice thing from a plant pathologist perspective regarding this disease is it's pretty easy to identify. So it has very distinct signs. It has these white fluffy mycelia and those hardened structures called sclerotia that I previously mentioned. But we want to avoid this, right? We want to get fungicide applications on at early flowering, because once you have these symptoms, you're already going to be experiencing yield loss on this particular plant. All right, so ultimately you have wilt and lodging as a result of this constricted vasculature of the constricted stem of the plant. So this is a difficult disease to manage for several reasons. For one, it has a wide host range of over 400 different broadleaf plants. So in addition to crop plants, weeds can harbor this disease as well. So weed control is actually an aspect of disease management for Sclerotinia. The sclerotia can persist in the soil for up to 5 years. So rotation can help to manage the disease in terms of reducing the amount of inoculum, but it's not going to be able to eradicate the disease. Chemical management can be effective, but as mentioned, timing is critical at those early flowering stages. And strong resistance is lacking in commercial cultivars. So all commercial cultivars that have resistance to Sclerotinia actually have tolerance. So even in lines that you think may be resistant, you may still see Sclerotinia in your field, but the Sclerotinia that you have may not be so severe that it kills the plant. So instead of having a single gene that has really strong resistance, all resistance to Sclerotinia is what's called quantitative. So there are several genes that have small effects in terms of building resistance to this particular disease. So resistance, however, is one of our most effective approaches to manage most any disease. And this is why some of my work and all the work that I'll be discussing today is focused on enhancing host disease resistance. In order to do this, we are taking two approaches in our first field season. One is to enhance white mold research capacity, so providing resources and tools for growers and researchers to be able to better study this disease. And we're exploring new avenues of disease management through soybean canopy architecture traits. All right, so in order to kind of launch our research program, one important aspect of being able to do Sclerotinia research is to have a representative panel of isolates. So we've been collecting isolates from throughout Minnesota on various crops, so different accessions of Sclerotinia from different fields in order to do work that uses these isolates and is representative of the population of Sclerotinia here in Minnesota. So we've collected 22 isolates this summer, and I can take this as an opportunity to say if you have Sclerotinia in your field, we would happily take samples in order to increase our isolate collection to be able to do work that's relevant to the characteristics of those particular isolates. And one reason this is important is that we know that aggressiveness differs between isolates. So what I mean by that is that different isolates cause more or less damage on soybean. So some of them may just have mild infections that don't girdle the stem, whereas others are going to girdle the stem and kill the plant. And this is important when we're screening for varieties because we may not be able to detect differences among those varieties depending on the isolate that's used. So we're working to develop a panel that we can use for screening germplasm. So this work was initiated by a graduate student in the lab, Xuan Fu. So as mentioned, we have a panel of 22 different isolates, and what we're noticing is that these isolates do differ in aggressiveness. So we measured lesion size on soybean between 24 and 124 hours after inoculating. So this is an undergraduate researcher, Ella. She's inoculating the petiole of the soybean plant with the fungus, where it's able to then progress down the petiole and infect or not cause severe infections on the plant. And lesion size was measured at these various time points. So again, we are noticing differences between isolates, and we still have additional isolates to screen. So we're also working to develop reliable nurseries for white mold, and I know that may be kind of a strange concept. You usually don't want to encourage white mold pressure in your field, right? But in order to conduct research, we need uniform and consistent pressure from white mold. So we know that this disease, the development of it, is environmentally dependent. It's favored by by cool, moist conditions, and these conditions may not occur every season, and we also have to have inoculum present in the soil for the disease to occur. So in order to conduct experiments, we are cold conditioning sclerotia and introducing them to the soil in various ways so that in this next season, we can screen cultivars and conduct experiments in the field. So in order to introduce inoculum, we use several different methods. Unfortunately, sunflower is very susceptible to sclerotinia. So we introduced a slurry of the mycelia of the fungus to the back of sunflower heads. This is Leslie stabbing the back of the floral head and inoculating it with the slurry of sclerotinia. And then the sclerotia fall from the flower into the soil. So this kind of simulates a natural infection in the field. We're also sprinkling lab-generated sclerotia into the field the fall before trials. So this fall, we added them to the soil, and then we'll also cold condition in the lab and spring apply them to the field. So the sclerotia have to cold condition in order to form those structures called apothecia that release the spores. We're comparing these with naturally infested plots. And we had good pressure this past summer, so we had successful infection of our sunflowers, and so we have lots of sclerotia to add to the soil in order to conduct trials next summer. So this summer, we're going to plant soybean here and evaluate them for disease incidence and apothecia. We also, I should mention, mist these so that there's a conducive environment for the disease. So the hope is to be able to improve our capacity to do effective white mold research in the future by having a reliable field infestation method. All right, so next I'll transition to the discussion of disease management through canopy architecture traits. So I should mention that this project is in collaboration with the soybean breeder at the university, Aaron Lorenz. So he has a diverse array of lines, over 100 different lines that vary in their architecture traits. So what I mean by that is leaf area index, branching angle, plant height, the time at which the canopy of the plants closes, so a variety of architecture traits. And we know that architecture can be important for avoiding infection. So we start to see apothecia appear in the field whenever we have a closed canopy. So whenever your row, the soil in your row is no longer visible. And this also corresponds with agronomics, so having a higher plant population, having smaller rows, all of these management strategies that lead to higher yields, unfortunately, also encourage sclerotinia in the field. So an idea is that potentially we can alter the architecture of the plant and combine that with genetic resistance for white mold and not have to change production practices in a manner that may negatively impact yield. So that would be the long-term goal of this work. And the reason that canopy closure is so important too is that the formation of these apothecia are very dependent on specific light conditions, and the wavelengths of light that they're sensitive to are within the UVB range. So as mentioned, Dr. Lorenz had over 100 different lines that vary in their architectural traits. These were planted both in Waseca and St. Paul. And they were evaluated for phenotypic traits in addition to canopy closure by drone. So they were evaluated from planting every week or two for canopy closure via drone images. And we've collected those data. We have not analyzed the drone data yet, but they have been collected. We then scouted for apothecia and disease and collected UVB measurements prior to flowering and at early flowering stages, which are those critical points for sclerotinia infection. In the field, we had a dry summer and we did not see sclerotinia in our field trials other than just a few plots. So this kind of goes back to my previous point about the need for having plots that have consistent and reliable white mold pressure. So we collected, though, an array of data on light in addition to plant architecture, and we're reducing the number of lines down to 10 or 20. that we can then investigate in the field next year in white mold nurseries where we're more confident that we'll have disease pressure. So we're also interested in screening these lines in the greenhouse to look at their genetic resistance to white mold. So really interested in combining architectural avoidance of disease with genetic resistance in order to enhance resistance to sclerotinia. So as a result of this work, a Sclerotinia sclerotiorem isolate collection has been developed and will continue to increase for future white mold work. We're also working to identify new breeding opportunities by enhancing disease escape with plant architecture traits and identifying candidate lines for breeding using these architecture traits. So we're also identifying lines with unique branching phenotypes that can lead to candidates for genetic identification of quantitative trait loci, so different loci that can again be used for high throughput breeding in order to have these architecture traits to escape disease. We're also working to improve white mold resistance screening through an isolate panel and improved nursery methods. And I think it's important to mention that this is a collaborative effort. So as a plant pathologist, I'm working together with the soybean breeding, and I think a lot of ideas can be generated from this collaboration. And we're training graduate students and undergraduate researchers in grower-driven applied research in plant pathology and agronomy. All right, so this is not the end of my presentation yet, but this is the end of my section. These are a few of the folks who have helped with the work. So Xuan Fu is working on isolate characterization. Alicia is a new graduate student who is working on the plant architecture project. And we have a couple of undergraduate students who have been in the lab since last spring, and I've been fortunate to have them stick on through the winter. And I'll take questions, I suppose, at the end. All right, so as mentioned, Ashish Ranjan could not be here today, so I am presenting the results from his initial field season. And his work is on screening soybean varieties to identify genetic resources for resistance against white mold. And again, this is in collaboration with the breeder, Aaron Lorenz. So the primary purpose of his work is to screen varieties for white mold resistance or potentially susceptibility in a naturally infested field environment. He's also screening them in the greenhouse and growth chambers to look at their genetic resistance in the absence of any potential escape mechanisms or environmental conditions. So in order to do this, he is working again in a naturally infested environment and in a growth chamber. So he's looking at 48 different soybean varieties. So these include lines from the breeding program at the University of Minnesota, in addition to a few commercial varieties that he can compare the results of the breeding lines to. So these were planted at the end of May, and this is a grower field in Faribault County at Johnson Farms. And this is a grower who had a known history of white mold in their field, and Ashish was able to observe white mold in his trials, so he was able to get good pressure. These were planted on 30-inch rows. He did mention that there was some mechanical damage from hand weeding, but they conducted stand counts and overall had good agronomics to be able to compare lines. So in order to compare the lines, he evaluated them for their disease severity, and this particular scale was used. So a rating of 0 had no infection, a rating of 1 had infection on the petiole, but it didn't progress to the main stem and cause a girdling lesion. A disease severity rating of 2 had an infection on the stem, and then 3 had a girdling of the stem, which is where you're going to see lodging and wilt and yield loss. So as mentioned, he did have white mold infections in the field, so he was able to compare these lines for their resistance to the disease. He also saw a large separation in the disease response of these lines. So those that had a low disease score or a low disease severity index are here on the left, and those that had high levels of infection are here on the right. And as mentioned, he also inoculated these in the growth chamber using the petiole inoculation method that I previously mentioned we're using in our work. So inoculating at the petiole with an auger plug that has the fungus, the mycelia of the fungus on it, so it can then progress down the petiole and cause infection. So in the greenhouse, again, he saw a large separation of resistance, some that had very small lesions that formed and others that had large lesions that were resulting in plant death. And he's evaluated 32 soybean lines so far, and this was at 10 days after inoculation. So this was the very end of the evaluation period, where at this point, if they were going to have girdling lesions that would result in the plant dying, it would be evident. All right, so through these two evaluation processes, he was able to identify some lines that had consistent susceptibility and consistent resistance to white mold, both in the field and in the greenhouse. So these can be used as sources of resistance. They can also be used as check lines to be able to compare commercial varieties and breeding lines to be able to understand whether they may be resistant or susceptible. So by evaluating soybean varieties both in a controlled environment and a field study, he identified a few that are highly resistant to white mold. And in the future, he's going to complete the growth chamber study using the rest of the soybean varieties. So he's evaluated 32 out of 48 thus far. He also needs to repeat his field evaluations to be able to have a reliable understanding of the results and of how susceptible versus resistant these lines are. And ultimately, these studies will pave the way to identify genes for resistance, and they can be used in breeding efforts in the future to enhance white mold resistance. This is Ashish's lab at the University of Minnesota, and our work is complementary, and ultimately we're aiming to really get at developing better resistance to this difficult to manage disease, because we know that it continues to be a yield limiting disease and it continues to be unpredictable. All right, great. And I think I have time for questions if there are any, a couple minutes. Yes. Yeah, so that's interesting because tillage can interact with sclerotinia in various ways. So the sclerotia are harbored in the soil. So tillage can, if it's a surface level tillage, can bring up new sclerotia. If it's a deep tillage, it could bury sclerotia. Also, residue can harbor sclerotia. So if you have residue on the surface of the soil, it's possible that it could have those sclerotia close to the surface of the soil. Another interesting research study that was published a few years ago had to do with roller crimped rye as well. So as mentioned, light is really important to the production of those apothecia. So in this particular study, having roller crimped rye on the surface of the soil, the apothecia weren't able to properly form because they didn't have the correct light conditions. So I think there are a lot of questions to explore with residue management and cover cropping management, and in general having different tillage practices or different layers of organic matter on the surface of the soil. So I don't have a conclusive answer for you, other than there are a variety of ways that tillage can interact or has the potential to interact with sclerotinia. But that's helpful to think about for future research questions.